Hello, ladies and gentlemen. I welcome each and every one of you to another session of a Daniel Hifos lecture. Today's lecture is intended to help facilitate your learning of the organic chemistry module of the essentials of the chemistry of life. This is the part 2 of a Dr. Hifos lectures on the chemistry of life. Gratitude for attending. Great. In the last lecture, the part 1, I introduced you to some essential points in the area of the chemistry of life with considerable emphasis on the inorganic component. I also did promise you that in the part 2, we will explore the organic chemistry aspect of the show. To begin with, let us answer the question, what are the differences between organic and inorganic substances or molecules? What are the differences between organic and inorganic substances or molecules? Ladies and gentlemen, inorganic substances usually have no carbon. Of course, the word usually suggests that there are exceptions, and in fact, there are exceptions. Carbon dioxide, carbonic acid, and calcium carbonate, for example, are inorganic substances with carbon in them. Often, inorganic substances contain positive and negative ions. They are usually small molecules and characterize non-living things to a comparatively larger extent. Examples of inorganic substances or molecules are oxygen, water, and carbon dioxide. Of course, you recall from the previous lecture that more than half of the body of the human being is made up of water. Another question is, what do you recall about oxygen and the composition of the weight of the human body? What do you recall about oxygen and the composition of the weight of the human body? I would be grateful if you could present your answer to this question as a comment on this video on the DH Media GH channel. In the meantime, what can we also say about the organic substances? Ladies and gentlemen, all of the organic substances have carbon. Most of them contain hydrogen. Many of them have oxygen or nitrogen, and all of them are covalently bonded. Yes, all of the known organic substances have carbon. Most of them contain hydrogen. Many of them have oxygen or nitrogen, and all of them are covalently bonded. In addition, organic substances or organic molecules or organic compounds are often large and associated with living organisms, including you and I, of course. Examples of organic substances are carbohydrates, lipids, nucleic acids, and proteins. Did you know it is the chemistry of carbon with atomic number 6 and chemical symbol of capital C which is responsible for the diversity of organic molecules which are found in living organisms, including humans, domestic animals, domesticated plants, pathogens, and disease vectors. Carbon can afford to form or to bond with as many as 4 other atoms. It can also bond with itself to produce both chains and rings. The fact that the nature of carbon allows for 4 bonds to be formed, coupled with the fact that it can have double, single, and triple bonds account for the formation of diverse organic molecules, and so the unity and diversity of life. Ladies and gentlemen, what is the point here? I have indicated that the fact that the nature of carbon allows 4 bonds to be formed, coupled with the fact that it can have double, single, and triple bonds, accounts for the formation of diverse organic molecules, and so the unity and diversity of living organisms. Did you know there are no two humans who have exactly the same fingerprints? And just like a human fingerprint, no two Siberian tigers have the exact same stripe pattern on their bodies. Can you imagine that? This is chemistry of life for you. Great. Ladies and gentlemen, each organic molecule has what we refer to as carbon chains. Yes, each organic molecule has what we refer to as carbon chains. The carbon chain of an organic molecule is referred to as its skeleton or backbone. Another vocabulary of interest is functional group. Perhaps you want to ask, what constitutes a functional group? Note that functional groups are clusters of specific atoms which are bonded to the carbon skeleton of the organic molecules with characteristic features and functions. Yes, functional groups are clusters of specific atoms which are bonded to the carbon skeleton of the organic molecules with characteristic structures and functions. The significance of the functional groups include the fact that they determine the chemical reactivity and polarity of organic molecules. The functional groups, their significance include the fact that they determine the chemical reactivity and polarity of organic molecules. Some of the functional groups which are explored in the world of chemistry of life are amino, carboxyl, hydroxyl, sulfhydryl, phosphate, and carbonyl functional groups. The carbonyl functional group may be the type found in an aldehyde or a ketone. Did you know the hydroxyl functional group occurs in alcohols, for example, methanol and ethanol? The amino functional group occurs in amines such as tryptophan. The sulfhydryl group is found in thiols such as ethane thiol. The phosphate is found in organic phosphate or the phosphorylated molecules. Yes, the phosphate is found in organic phosphate or what we call the phosphorylated molecules. The carboxyl functional group is also found in organic acids such as acetic acid, formic acid, and ethanoic acid. The carboxyl functional group is found in an organic acid such as acetic acid, formic acid, and ethanoic acid. Again, the aldehyde type of the carbonyl functional group is found in an aldehyde like formaldehyde and other aldehydes that are known to science. Lastly, the ketone type of the carbonyl group occurs in ketones such as the acetone. The next question is of interest to you in particular, and that question is, what is the significance of each of the functional groups in the chemistry of life? The hydroxyl functional group is polar in nature and forms hydrogen bond. It is found in sugars and some amino acids. The carbonyl functional group is also polar. Both the types, which are found in aldehyde and ketones, occur in sugars. The carboxyl functional group is acidic and occurs in amino acids and fatty acids. The amino functional group is polar, basic, and forms hydrogen bonds. It is found in amino acids. The sulfhydryl functional group forms what we call disulfide bonds and occurs in some amino acids. Lastly, the phosphate functional group is polar, acidic, and occurs in nucleotides and phospholipids. Ladies and gentlemen, take note of the fact that it is the diversity in the carbon skeleton of organic molecules and the attached functional groups which make it possible for such molecules to demonstrate diversity in chemical properties. Furthermore, it is the chemical properties of a molecule which dictate how that molecule interacts with other molecules and the role it plays in the cell of living organisms. Yes, it is the chemical properties of a molecule which dictate how that molecule interacts with other molecules and the role it plays in the cell of living organisms. Some functional groups are said to be hydrophilic in nature, whereas others are known to be hydrophobic in nature. What is the difference? The difference is that the hydrophilic substances or functional groups interact with water by dissolving in water and or forming hydrogen bonds with water molecules. On the other hand, the hydrophobic functional groups or substances or molecules do not interact with water because they are nonpolar in nature. I hope you remember what is meant by polar and nonpolar covalent molecules. Indeed, the four important classes of biomolecules which have been found in the cells of living organisms, including those of humans, are carbohydrates, nucleic acids, lipids, and proteins. Earlier on, we mentioned these as examples of organic molecules. Yes, these are called biological molecules or simply biomolecules. They usually have many repeating units. Each of the repeating units is a monomer. A molecule which is made of monomers is called a polymer. That is, that molecule has many parts. Amino acids as monomers, for example, are joined together to form a protein, and that protein is a polymer. Yes, amino acids are monomers, and they are joined together to form a protein. And that protein which is formed, the product, is a polymer. Ladies and gentlemen, the largest carbohydrates are known as polysaccharides. Such polysaccharides are polymers of simple sugars which are referred to as monosaccharides. A carbohydrate, which is composed of two monosaccharides, is said to be a disaccharide. The nucleic acids are polymers of subunits or monomers called nucleotides, whereas the polypeptides of proteins are polymers of amino acids as indicated earlier on. Furthermore, the monomers or subunits of lipids are glycerol and fatty acids. Dear ladies and gentlemen, polymers are produced through the joining together of monomers. For each bond formed in the course of hydrolysis, a molecule of water is added. For each bond formed in the course of hydrolysis, a molecule of water is added. On the other hand, for each bond formed in a dehydration kind of reaction, a molecule of water is removed. In other words, the term hydrolysis reaction refers to the splitting of a bond by the addition of water molecule, with the hydrogen ion being presented to one of the molecules involved and the hydroxide ion being accepted by the other molecule involved in the reaction. On the other hand, dehydration reaction has been defined as a type of reaction which leads to the formation of a covalent bond with the associated loss of a water molecule. The term carbohydrate literally means hydrated carbon. In other words, carbohydrate is made up of a carbon to which water has been added. Carbohydrates include monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Thus, the term carbohydrate is defined to include both the monomers such as glucose and ribose, and the polymers such as starch, glycogen, and cellulose. Yes, when you define carbohydrates, okay, it should include both the monomers such as glucose and ribose, and the polymers such as starch, glycogen, and cellulose. Glucose is the immediate energy source of cells. Glucose is the immediate energy source of cells. Starch, glycogen, cellulose, and other polysaccharides are polymers of glucose. Starch, glycogen, cellulose, and other polysaccharides are polymers of glucose. Glycogen in animals, including human beings, is an energy storage compound. So glycogen is an energy storage compound. Indeed, carbohydrates are the major source of energy for your body as a human being. They also offer to you some important body burden materials. Yes, carbohydrates are source of important body burden material. They contain carbon, hydrogen, and oxygen, and often twice as much hydrogen as oxygen, as you can see in the case of glucose. Glucose has the formula C6H12O6. In other words, you can find 6 of the oxygen and 12 hydrogen in glucose, and that is why I have indicated that, yes, carbohydrates contain carbon, hydrogen, and oxygen, and often twice as much hydrogen as oxygen. Ladies and gentlemen, each monosaccharide, when I say monosaccharide, I mean one sugar, okay? So each monosaccharide has, let's say, 3 to 7 carbons, and maybe a straight chain or a ring. Each monosaccharide may have 3 to 7 carbons and may be a straight chain or ring. Examples are glucose or dextrose, fructose, galactose, and ribose. Each disaccharide contains 2 sugars with 6 carbon units. Examples of disaccharides are sucrose, or the table sugar, which contains glucose and fructose; lactose, or milk sugar, which is made up of glucose and galactose; and maltose, or malt sugar, which contains 2 molecules of glucose. In other words, maltose or malt sugar consists of glucose plus glucose. Okay, glucose plus glucose. Ladies and gentlemen, polysaccharides are long chains of simple sugars. Yes, we have talked about this severally, that polysaccharides are long chains of simple sugars. They are highly branched. In the human body, it is only the chains of glucose which are important. Yes, in your body as a human being, it is only the chains of glucose which are important. Glycogen is a polysaccharide and is called animal starch, and it's actually found in human beings. So you have glycogen in your body. Glycogen is stored in skeletal muscle and an organ called the liver. Yes, so you store your glycogen in your skeletal muscle and also your liver. Did you know glycogen can also be broken down into glucose when energy is required? Yes, glycogen can also be broken down into glucose when required for energy. Starch from grains and potatoes are of plant origin. Human beings, including you and I, we are able to digest and use starch. Yes, we are able to digest and use starch. But note that it's really not every carbohydrate that we can afford to digest. Did you know there are certain 2 unique polysaccharides, namely cellulose and chitin, which are also important in the world of chemistry of life? The chitin contains the amino functional group. The chitin contains the amino functional group. It is found in exoskeleton of insects and other arthropods. It is not digestible by human beings. What about the cellulose? The cellulose also forms straight chain bonded together by hydrogen bonds. The cellulose forms straight chains bonded together by hydrogen bonds. Although you cannot afford to digest them, they add bulk to your meal. Yes, we human beings, we are unable to digest cellulose, but they still have an advantage, or they provide an advantage by adding to the bulk of our meals. Great. Ladies and gentlemen, let's shift our attention to lipids. Did you know lipids have carbon, hydrogen, and oxygen? They have lesser number of oxygens than carbohydrates. Yes, lipids have lesser number of oxygens than carbohydrates. They are insoluble in water. Okay, lipids are insoluble in water. They are large molecules and include diverse compounds. Yes, there's a lot of diversity among the lipid molecules. They are called fats and waxes when they are solid, and when they are liquid, they are usually referred to as oils. Yes, lipids are called fats and waxes when they are solid, and oils when they are liquid. Indeed, lipid molecules include fats, oils, waxes, phospholipids, and steroids. Lipid molecules include fats, oils, waxes, phospholipids, and steroids. They are used for energy. They are used for energy. Of course, it's not only energy. We'll talk about some other uses of lipids like the steroids shortly. Yes. Lipids also serve in terms of insulation and protection. Lipids, they also serve in terms of insulation and protection and as part of cell walls. Some hormones are also lipids. Yes, the steroids, for example, include hormones. And so some hormones are also lipids. Fats and oils allow long-term energy storage. Fats and oils allow long-term energy storage. Some differences between fats and oils are that fats are typically of animal origin and are solid at room temperature, but oils are of plant origin and are liquid at room temperature. Ladies and gentlemen, each lipid is made up of 1 glycerol and 3 fatty acids. Each lipid molecule is made up of 1 glycerol and 3 fatty acids. Accordingly, they are called triglycerides. Their glycerol backbone is the same, whereas the fatty acid change. Their glycerol backbone is the same or remains the same, whereas the fatty acids change. The glycerol portion has hydroxyl groups. The glycerol portion of the lipid, the glycerol portion of the lipid has what we call hydroxyl groups, the OH. And the hydroxyl group, there makes them polar. So the glycerol portion is actually polar. So let me put it this way. The glycerol portion of lipids have, I beg your pardon, the glycerol portion of lipid has hydroxyl groups which are polar. Therefore, this portion of the lipid is hydrophilic. The glycerol portion of the lipid is hydrophilic. Since the methyl group is nonpolar, fatty acids are hydrophobic. Since the methyl group is nonpolar, fatty acids are hydrophobic. Even though both glycerol and fatty acids possess polar groups, fat and oils are nonpolar, and that basically explains why they are insoluble in water. Even though both glycerol and fatty acids possess polar groups, fat and oils are nonpolar, and that basically explains why they are insoluble in water. Ladies and gentlemen, fats tend to have saturated fatty acids. Fats tend to have saturated fatty acids, and oils tend to have unsaturated fatty acids. Saturated fatty acids lack carbon-carbon double bond, but unsaturated fatty acids possess double bond in their hydrogen or their hydrocarbon chain. So saturated fatty acids lack carbon-carbon double bonds, but unsaturated fatty acids possess double bond in their hydrocarbon chain. The double bond produces a kink in the unsaturated fatty acids, and it is that which explains the liquid nature of oils at room temperature. Studies have shown that Rodents and birds in the temperate regions of the world have unsaturated triglycerides in their feet, and that helps to keep them from freezing. Ladies and gentlemen, recall the fact that saturated fats have single covalent bonds between carbons of fatty acids. Remember the fact that saturated fats have single covalent bonds between carbons of fatty acids or fatty acids. That is to say that they are carbons saturated with hydrocarbons. Okay, so saturated fats which have single covalent bonds between carbons of fatty acids, okay, are said to be carbons which have been saturated with hydrogens. Let me take it again. Recall the fact that saturated fats have single covalent bonds between carbons of fatty acids. And that is to say that they are carbons saturated with hydrogens. Such fats tend to be solid at room temperature, and they include animal fat, butter, cheese, coconut, eggs, meat fat, milk, and palm oil. Yes, our own palm oil. Ladies and gentlemen, consuming and storing high levels of saturated fats could cause one to suffer a biomedical condition called atherosclerosis or arteriosclerosis by increasing the cholesterol the body makes. Yes, when you choose to consume and store high levels of saturated fats, okay, such a situation could cause you to suffer a biomedical condition called atherosclerosis or arteriosclerosis. And it does that by increasing the cholesterol your body makes. This medical condition basically refers to an obstruction of the victim's blood vessels. Yes, your blood vessels will be obstructed when you suffer atherosclerosis or arteriosclerosis. Now, do a simple search after this lecture and use your own words to define atherosclerosis or arteriosclerosis. Leave your definition as a comment on the video on the DH Media GA channel on YouTube. Ladies and gentlemen, let's shift our attention to the unsaturated fats. The unsaturated fats are characterized by fatty acids which contain one or more double covalent bonds between carbons of fatty acids. The unsaturated fats are characterized by fatty acids which contain one or more double covalent bonds between carbons of fatty acids. That is, their carbons are not saturated with hydrogens. Okay, so saturated fats, their carbons are saturated with hydrogens, whilst unsaturated fats have carbons which are not saturated with hydrogens. The unsaturated fats tend to be liquid at room temperature, and they include cooking oils. Of course, you know about the olives or olive oils. You know about the avocados, seeds, nuts, peanuts. Okay, they are examples of unsaturated fatty acids, and in actual fact, they are said to be monounsaturated. I repeat, olives, olive oils, avocados, seeds, nuts, and peanuts are said to be monounsaturated. If we have monounsaturated, then we can also afford to have polyunsaturated. What are they? Polyunsaturated fats can be found in corn, soybean, fish, flaxseed oil, okay, sunflower, and walnuts. Yes, polyunsaturated unsaturated fats can be found in corn, soybean, fish, flaxseed oils, flour, like sunflower, walnuts, and flax seeds. Within polyunsaturated fats are also two essential fats. Within polyunsaturated fats are also two essential fats, namely omega-3 fatty acids and omega-6 fatty acids. So there are two essential fats, within polyunsaturated fats, and they are omega-3 fatty acids and omega-6 fatty acids. I'm sure you should be interested in finding out much about omega-3 fatty acids and omega-6 fatty acids. Note that the polyunsaturated fats have more than one double or triple bonds. The polyunsaturated fats have more than one double or triple bonds. I want you to also conduct research and find out the significance of having more than one double or triple bonds in fatty acids. And of course, I have given you the clue already, but you should try and do more research on such a thing. Have you heard, ladies and gentlemen, unsaturated fats can be transformed into saturated ones just by having hydrogens added to them? Yes, you can afford to transform unsaturated fats into saturated ones just by adding hydrogens to them. The process is called hydrogenation, and the products are referred to as being hydrogenated fats. Did you know there are many processed foods which have hydrogenated fats in them? Yes, there are many processed foods with hydrogenated fats in them. In actual fact, they truly taste very good, very delicious. But the bitter truth is that they are truly bad for you and also your loved ones. Ladies and gentlemen, at this point, let us shift our attention to the phospholipids. Phospholipids are one of the classes of lipids whose molecule has a hydrophilic head having a phosphate group and two hydrophobic tails which are derived from fatty acids and joined together by an alcohol residue. I repeat, phospholipids are one of the classes of lipids whose molecule has a hydrophilic head having a phosphate group and two hydrophobic tails which are derived from fatty acids and joined together by an alcohol residue. The alcohol residue under consideration is usually a glycerol molecule. The alcohol residue here is usually a glycerol molecule. In other words, in a lipid called phospholipid, one of the three fatty acids is substituted by a phosphate group. In a lipid called phospholipid, one of the three fatty acids is substituted by a phosphate group. Remember, we said that fatty acids are made up of one glycerol and three fatty acids. In the case of the phospholipids, one of the three fatty acids is substituted by a phosphate group. So it means it has two instead of three fatty acids. Phospholipids are important parts of all plasma membranes. Yes, we are talking about the importance or significance of phospholipids. They are important part of all cell or plasma membranes. They are able to form lipid bilayers due to their amphiphilic characteristic nature. Yes, they are amphiphilic in nature. Probably you would want to do some research so you can afford to define vividly what is meant by the amphiphilic nature of phospholipids. In the eukaryotic organisms, ladies and gentlemen, note that in the eukaryotic organisms, cell membranes also contain another class of lipid called sterol, which is interspersed among the phospholipids. In the eukaryotic organisms, cell membranes also contain another class of lipid called sterol, which is interspersed among the phospholipids. Together, they provide fluidity in 2 dimensions and mechanical strength against rupture. Together, they provide fluidity in 2 dimensions and mechanical strength against rupture. Indeed, phospholipids play key structural and metabolic roles in cells. They include lecithin, cephalin, plasmalogens, and cardiolipin. Phospholipids play key structural and metabolic roles in cells, and they include lecithin, cephalin, plasmalogens, and cardiolipin. Plasmalogens are found in the brain and heart, whereas the cardiolipin has been associated with the heart. The word cardiology. Cardio has to do with the heart. The lipids called steroids possess 4 fused rings, which are designated A, B, C, and D, and are arranged in a specific molecular configuration. Yes, at this moment, we are talking about the lipids called steroids. And we have indicated that they possess 4 fused rings, which are designated A, B, C, and D, and are arranged in a specific molecular configuration. They have 2 key biological functions. That is, they are key part of cell membranes, which change fluidity, and they serve as signaling molecules. They are key parts of cell membranes which change fluidity, and they serve as signaling molecules. Apart from cholesterol, sex hormones such as estrogen, like estradiol, and testosterone are steroids. So estrogen is a steroid, and testosterone is a steroid. Did you know the estradiol helps in the regulation of female reproductive cycles like the estrous cycle and menstrual cycle? The estradiol helps in the regulation of female reproductive cycles like the estrous cycle and menstrual cycle. It is responsible for the development of female secondary sexual characteristics, including the enlargement of the breast, widening of the hips, and a female pattern of body fat distribution. It is also crucial in the development and maintenance of a female reproductive tissue, such as those associated with the mammary glands, uterus, and the vagina during the puberty stage, as well as in adulthood and pregnancy. Yes, the estradiol is crucial in the development and maintenance of female reproductive tissues, such as those which are associated with the breast or the mammary glands, uterus, and the vagina during the puberty stage, as well as in adulthood and in pregnancy. This hormone also has critical impacts in other tissues such as the brain, bone, fat, and the skin, and even the liver. Yes, this hormone has critical impact in other tissues such as the brain, bone, fat, liver, and the skin. Now, what can we say about the waxes? What do you want to learn about the waxes? Waxes are another group or a second group of neutral lipids. Okay, waxes are judged a second group of neutral lipids. They are important physiologically, albeit they are a minor constituent of biological systems. Yes, although waxes are a minor constituent or component of biological systems, they are important in physiological terms. They basically comprise of a long-chain fatty acid bonded through an ester oxygen to a long-chain alcohol. Yes, waxes are basically comprised of a long-chain fatty acid bonded through an ester oxygen to a long-chain alcohol. They are fully water-insoluble molecules and characteristically solid at biological temperatures. Waxes are fully insoluble molecules and characteristically solid at biological temperatures. They are insoluble in water or completely insoluble in water. For those of you who are interested in biological sciences and even natural resources, take note of this. First, the biological scientist. For you, the biological scientist, I know that you will be glad to note that waxes serve as water repellent on the leaves of some plant species, on feathers of bird species, and on the cuticles of some insect species. Again, they serve as energy storage substances in microscopic aquatic plants and animals called phytoplankton and zooplankton, respectively. Yes, waxes serve as energy storage substances in microscopic aquatic plants called phytoplankton and microscopic aquatic animals called zooplankton. In addition, they serve as energy storage substances in higher ranking members of the aquatic food chain. Waxes serve as energy storage substances in higher ranking members of the aquatic food chain. According to some scientific authorities, plankton, be they phyto or zooplankton, employs the biological synthesis of waxes to regulate their buoyant density and therefore their depth in the oceanic world. Wow! Scientists have found out that plankton employs the biological synthesis of waxes to regulate their buoyant density and therefore their depth in the oceanic world. They're able to tell their depth, or they're able to regulate their depth in the oceanic world. For those of you who are interested in the natural resources, it has been reported that one of the key sources of petroleum in deep sea deposits or deep sea sediment is the deposition of wax-rich dead plankton over so many years. Yes, it has been reported, has been found that one of the important sources of petroleum in deep sea deposits or sediments is the deposition of wax-rich dead plankton over so many years. Lastly, take note of the fact that both fishes and whales have been reported to store large amounts of waxes in their bodies. On proteins, let us indicate that proteins perform many functions in cells and living organisms, including the human being. Such functions include those related to defense or immunity, metabolism, motion, regulation, transport, and support. Proteins, polymers, Proteins polymers are made from amino acids. In other words, proteins are polymers of amino acids. Did you know there's something we call peptide? What is it? A peptide refers to a short string of 2 to 50 amino acids which are formed through a condensation reaction and covalent bond or bonds. Okay, a peptide refers to a short string of 2 to 50 amino acids which are formed through a condensation reaction and covalent bond or bonds. In other words, peptides are chains of amino acids. A peptide has smaller version. I beg your pardon. A peptide, a smaller version of a protein, probably offers considerable pro-aging support, anti-inflammatory or muscle building functions or properties. Yes. A peptide, a smaller version of a protein, probably offers considerable pro-aging support, anti-inflammatory or muscle building functions. One difference between peptides and proteins is that peptides contain two, let me frame it this way. One major difference between peptides and proteins is that peptides contain between 2 and 50. Peptides contain between 2 and 50 amino acids, and proteins contain 50 or more amino acids. Okay, so one way you can differentiate between peptides and protein is that peptides contain or have they have between 2 and 50 amino acids, and proteins contain 50 or more amino acids. Peptides contain between 2 and 50 amino acids, and proteins contain 50 or more amino acids. Besides, peptides tend to be smaller and less well-defined in structure compared to proteins. Peptides tend to be smaller and less well-defined in structure compared to proteins. Unlike peptides, proteins can adopt complex forms including secondary, tertiary, and quaternary structures. Furthermore, peptides may be oligopeptides with as few as 2 to 20 amino acids, or polypeptides with many amino acids. That is, a polypeptide is a relatively longer chain of amino acids joined through peptide bond. A polypeptide is a relatively longer chain of amino acids joined via peptide bonds. On the other hand, proteins are one or more polypeptides joined together, and that makes proteins basically very large. Okay, or we can even refer to proteins as very large peptides. Yes, proteins are one or more polypeptides joined together, and that makes proteins basically very large peptides. According to some authorities, the term peptide should be used to refer specifically to oligopeptides or relatively short amino acids. According to those authorities, the term polypeptide should be used for proteins or chains of 50 or more amino acids. Did you know there are 20 different amino acids in cells, and such amino acids vary only by their R group? Note also that presence or absence of polarity is a key aspect of the R group. A polypeptide possesses up to 4 levels of structure, namely primary, secondary, tertiary, and quaternary levels of structure. The primary level is the sequence of the amino acids which differs between polypeptides. The primary level is the sequence of the amino acids which differs between polypeptides. The secondary level is characterized by an alpha helixes or alpha helixes and beta pleated sheets which are held in place by means of hydrogen bonding between amino acids along the polypeptide chain. Yes, the secondary level is characterized by alpha helices and beta or pleated sheets which are held in place by means of hydrogen bonding between amino acids along the polypeptide chain. The tertiary level is the final folding of the polypeptide, and it is held in place by bonding and hydrophobic interaction between R groups. The tertiary level is the final folding of the polypeptide, and it is held in place by bonding and hydrophobic interactions between R groups. Proteins with more than one polypeptide possess a quaternary level of structure as well. Proteins with more than one polypeptide possess a quaternary level of structure as well. Indeed, all proteins have a primary structure. All proteins have a primary structure. Both globular and fibrous proteins have a secondary structure. They are either helixes, as found in keratin. Let's say keratin that we find in hair, horns, and hoof, in addition to collagen, or they may be pleated sheets as found in silk, which is made by spiders and silkworm. In other words, the structures under consideration, okay, we could find them in keratin of hairs, horns, and hoof, in addition to collagen, or pleated sheets as could be found in silk, which is made by spiders and silkworms. Furthermore, globular proteins always possess a tertiary structure, and most of them possess a quaternary structure as found in hemoglobin and various enzymes like RNAs. Yes, so it means hemoglobin has a quaternary structure, and then an enzyme like the RNAs also has a quaternary structure. Did you know, we've been talking about structures. Okay, listen to this. The proteins of a cell determine its structure and function. The proteins of a cell determine its structure and functions. Again, the shape of an enzyme is key to its function. And you know, an enzyme is a protein. Okay, in other words, the shape of a protein is key to its function. Did you know both high temperatures and a change in pH can denature proteins? Both high temperatures and a change in pH denature proteins. And those proteins, as I've indicated, include enzymes. And such a thing will cause them to lose their shape and hence their function. When a protein loses its shape, it will also lose its function. When a protein loses its shape, it will also lose its function. Now let's turn our attention to the nucleic acid. What do we have to say about the nucleic acid? The nucleic acids, namely deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are polymers of nucleotides. There is the likelihood of diversity due to the fact that the nucleotides can be in any order. The nucleotides, which are the subunits of the nucleic acids, okay, their arrangements can be in any order. Each of the nucleotide is made up of 3 components. When you take any nucleotide, it has 3 parts, 3 components. The 3 components of nucleotides are a pentose or 5-carbon sugar, a phosphate or phosphoric acid, in addition to a nitrogen-containing base or a nitrogenous base. In other words, a nucleotide is made up of a pentose sugar, a phosphate, and a nitrogen-containing base. Ladies and gentlemen, note that DNA contains the pentose sugar called deoxyribose. In other words, in that pentose sugar, an oxygen is absent. That's why we say it is deoxyribose, and that's why DNA has the name deoxyribose. Deoxy. So DNA contains the pentose sugar called deoxyribose. It is the genetic material which stores information for its own replication and for the order in which amino acids are supposed to be sequenced in proteins. With the help of what we call the messenger RNA or mRNA, DNA specifies protein synthesis. Ladies and gentlemen, the DNA contains phosphate, the pentose sugar called deoxyribose, and nitrogen-containing bases. The DNA is a double-stranded helix in which adenine A always pairs with thymine T, and cytosine C always pairs with guanine G, and the pairing or the bonding always takes through hydrogen bonding. Okay, so DNA is a double-stranded helix in which adenine always pairs with thymine and cytosine always pairs with guanine through or via hydrogen bonding. This is referred to as complementary base pairing. Complementary base pairing. So, irrespective of the quantity or order of any particular base pair, the number of pyrimidine bases always equals the number of purine bases. The pyrimidine bases are cytosine and thymine, and the purine bases are adenine and guanine in the case of DNA. Let us turn our attention to the RNA. The RNA contains phosphate, the sugar called ribose instead of deoxyribose, and the bases are adenine, uracil instead of thymine, cytosine, and guanine. In other words, both DNA and RNA have 4 of the bases. 2 of them are purines in the case of DNA, and the other 2 are pyrimidines. The pyrimidines that you can find in DNA are cytosine and thymine, and the purines are adenine and guanine. In the case of the RNA, okay, the bases are adenine, uracil, cytosine, and guanine. Okay, so it means that what is absent in the case of the RNA is the thymine, which has been replaced with uracil. The RNA is single-stranded. There are 3 types of RNA which are produced in the nucleus. They are messenger RNA, mRNA, ribosomal RNA or rRNA, and transfer RNA or what we call tRNA. The rRNA is made in the nucleolus of the nucleus. The nucleolus is a dark region of chromatin where RNA joins with proteins to form the subunit of organelles called ribosomes. These organelles are small membrane-bound bodies in the cytoplasm where protein synthesis takes place. Yes, the nucleolus is a dark region of chromatin where RNA joins with protein to form the subunit of organelles called ribosome. So it is rRNA which joins with protein, which joins with proteins. The rRNA joins with proteins to form the subunit of organelles called ribosomes. Now, the ribosomes can be described as being small organelles and that they are membrane-bound bodies and they occur in the cytoplasm. And it is in the cytoplasm that protein synthesis takes place. Anyway, I would like you to conduct more research and get some more information about the ribosomes, whether they are always in the cytoplasm or they occur somewhere else, whether they occur singly or as groups. Conduct some research and leave your answer or the outcome of your research as a comment on this video. Ladies and gentlemen, as noted earlier, mRNA is an intermediary in the process involved in protein synthesis. It carries information from DNA regarding the amino acid sequence in a protein. But what about the tRNA, the transfer RNA? The tRNA takes part in the assembly of amino acids during photosynthesis. So yes, we talk about 3 types of RNA: mRNA, rRNA, and tRNA. And we have indicated the functions of each of these RNAs. We've also indicated that the RNA, the rRNA is made in the nucleus of the nucleus. And we've indicated that the nucleolus is actually a dark region of chromatin where the rRNA, which is made in the nucleus, joins with protein to form the subunit of organelles called ribosomes. So you see that the function of the rRNA, the ribosomal RNA, lies in the formation of ribosomes. And you know that the ribosomes are for photosynthesis. You've also taken note of the fact that mRNA is an intermediary in the process involved in protein synthesis. And it is such that it carries information from DNA regarding or concerning the amino acid sequence in a protein. You've also taken note of the fact that the tRNA, the transfer RNA, takes part in the assembly of amino acids during protein synthesis. Ladies and gentlemen, did you know there is something we call ATP, the adenosine triphosphate, ATP. The adenosine triphosphate, the adenosine triphosphate, ATP, the adenosine triphosphate is also a nucleotide in which adenosine is made of adenine and ribose. The ATP is the energy currency, of cells. The ATP has been referred to as the energy currency of cells. The triphosphate stands for, you say ATP, adenosine triphosphate. What is the meaning of the triphosphate? The triphosphate stands for the 3 phosphate groups which are attached together and to the pentose sugar called ribose. The triphosphate stands for the three phosphate groups which are attached together into the pentose sugar called ribose. The reason ATP is a high energy molecule is that the last two phosphate bonds which it possesses are not stable, and so they are easily broken down. In cells, the hydrolysis of ATP to adenosine diphosphate plus phosphate releases energy. This energy is employed by cells to make a product or do any other type of metabolic activity. Ladies and gentlemen, all too soon, we have come to the end of today's presentation. Do well to ask your questions as a comment on the video on the YouTube, and I will surely answer each and every one of them. Thank you very much for participation or your participation in this lecture. I will see you again in another lecture where we'll be talking about cell structure and function. Bye.